Now the next step in this whole process of getting energy out of carbohydrates is to go over to the electron transport chain. Remember how I told you guys that in the mitochondria, there's an outer membrane, an inner membrane, and there's the matrix of the mitochondria. I drew it out for you in a previous video. And I told you guys when we were doing glycolysis and in the transition reaction between glycolysis and the citric acid cycle and in the citric acid cycle that we had to pay attention to all the NADHs that were being produced. And in the Krebs citric acid cycle, also the FADH2s. So all of those NADHs and FADH2s are all now, think about it, they're all in the matrix of the mitochondria. They all came through, they're in the matrix, and then they are going to be depositing their hydrogen ions and electrons into those carriers of the electron transport chain. Okay, now the process itself is actually pretty straightforward, but there are lots and lots of complicated steps. I'm going to oversimplify it for you guys, and you just need to know the basics. You don't need to know every single detail of every single step that is in the book or that OWL talks about. You just need to know the basic general thing of what's going on. Okay, so the electron transport chain is for, actually you have this in your book. You might want to print it out so you could take a look at it, or just open up your book and have it in front of you. So basically 4 major molecules. These are big protein molecules that are embedded into the inner membrane of the mitochondria. So what I have over here is the outer membrane, the intermembrane space, and the inner membrane. So remember how I had the mitochondria like this, outer membrane, inner membrane, matrix. So it's like I took a section and expanded it over here. Outer membrane, intramembrane space, the space between the two membranes, and the inner membrane. And then the matrix is down over here. Okay, so you have these 4 big protein molecules that are made up of many different things. And they are, we can just call them complex 1, complex 2, complex 3, and complex 4. So these ones, the ones that have the Roman numeral 1, Roman numeral 2, Roman numeral 3, Roman numeral 4. Those don't move, okay? They are embedded in the inner membrane. There are 2 mobile carriers of electrons. That's coenzyme Q. This one's right here between, it's right next to complex 2. And it can carry electrons from 1 to 2 and over to 3. And then there's cytochrome C right over here between 3 and 4. So coenzyme Q and cytochrome C move. The other complexes 1, 2, 3, and 4 don't move. They are embedded in the inner membrane. At the end of the electron transport chain, there's an oxygen. Oxygen is right next to complex number 4, okay? None of this would be possible if oxygen is not there. That's why you can't do any of this metabolism unless the cell is aerobic. If it's not aerobic, you can't get into the mitochondria and do any of this stuff, okay? Now, so what is actually going on? The NADHs and the FADH2s that we've been accumulating in all of the steps so far come right here. They're in the matrix and they deposit their hydrogen ions and electrons. Remember, a hydrogen atom is an H plus and an electron. So they deposit their hydrogen ions and their electrons right here. So the NADHs deposit their hydrogen ions and electrons right to complex number 1. And what happens is that complex 1 will pump the hydrogen ions into this intermembrane space. The FADH2 deposits it right here in complex number 2, pumps it into this intermembrane space. And we can pump also in complex 3. So we've got complex 1, complex 2, complex 3 pumping hydrogen ions into this intermembrane space. So now we have a whole bunch of hydrogen ions here. Okay, what happens to the electrons? The electrons get transported from one complex to the other Okay, so some hydrogen ions are still over here, electrons are getting transported over here. So during all of this, once the electrons and the hydrogen ions reach oxygen, this is what's going to happen over here. Water is going to be produced. So an O2 plus 4 hydrogen ions plus 4 electrons produces 2H2O. What do you need to know? You need to know that oxygen is right at the end of the electron transport chain, near complex number 4. The hydrogen ions and the electrons, not all hydrogen ions, most of the hydrogen ions are being pumped into this intermembrane space, but some hydrogen ions and electrons go over to the oxygen through the complexes. How do they go? How do the electrons go over? These are all redox reactions. Things are getting oxidized, reduced, oxidized, reduced. Anyway, there's a constant movement of the electrons through all these coupled reactions, all these redox reactions, and eventually it gets to the oxygen. And the oxygen will pick up the hydrogen ions and the electrons and get turned into water. Remember how we said that you're producing 6 moles of water from 1 mole of glucose? There's some water being produced. So keep going and you get your 6 waters. Okay, now what becomes of all these hydrogen ions? All these hydrogen ions start to accumulate, accumulate, accumulate. What happens when you have a lot of hydrogen ions in a specific location? The pH goes down. Right. Now, the term gradient means that you have more of something on one side of a membrane than on the other side. So you have something called a proton gradient right now. I'm going to move this guy. Proton gradient. What's a proton gradient? You have more protons in this intermembrane space than you do in the matrix. What's a proton? It's just a hydrogen ion, okay? Remember, a hydrogen atom is 1 proton, 1 electrons, no neutrons. So if it's a hydrogen ion, an H+, it lost its electron, all that's left is a proton. So that's why we call hydrogen ions protons sometimes. Right, you have a proton gradient. You have more protons in this intermembrane space than over here. There is also, I think your book calls it an electrochemical gradient or something like that. Yeah, electrochemical. Yeah, they call it an electrochemical proton gradient. More H pluses on this side than on this side. Okay. Just think of it as a dam. Like if you have a dam and you have a lot of water on one side, and then you open up the dam, all the water flows to the other side from a higher concentration to a lower concentration. Well, this over here, this F0, F1 protein over here, is, think of it as that opening of the dam. When your hydrogen ions reach a specific level, a specific pH in this intermembrane space, this F0, F1 protein over here opens up the channel and allows them to flow back into the matrix. Why is this important? All that energy, all that kinetic energy of all those protons flowing back into the matrix is then captured. You know how energy can't be created or destroyed, but it can be transferred? Well, all that energy is then transferred, and with the help of the enzyme ATP synthase, it's enough energy to get, see, an ADP and an inorganic phosphate are sitting right here. So when all these protons flow back into the matrix, this energy is captured, and you can create high energy bonds and turn ADP and inorganic phosphate into ATP. This method of making ATP is called oxidative phosphorylation. Oxidative phosphorylation. And it only occurs in the electron transport chain because of this proton gradient, okay? How did we get the proton gradient? Remember, NADHs and FADH2s dump their protons and their electrons onto these complexes. The complexes pumped the protons into the intermembrane space. The intermembrane space had a huge accumulation of protons. Once it reached a certain level, this F0, F1 protein opened up, allowed all the protons to flow back into the matrix. All that energy was captured. ATP synthase enzyme catalyzed the reaction of adding a high energy phosphate onto ADP. Why is it called oxidative phosphorylation? Because you have to have oxygen for all of this to occur. Okay? All right. We will finish up the story in the next video.